I'm going to talk about macrophage migration inhibitory factor. This is like known as MIF or MIF, and like the importance of this molecule or how this molecule been implicated in several inflammatory and autoimmune diseases, and I'm going to focus right now on the role of MIF in sepsis. And I'm going to highlight like the subject which I'm going to cover in my talk, which will be how MIF act as a late mediator of sepsis, and as well as like the beneficial effect of inhibiting MIF using like a small molecule that we designed in our lab at the Feinstein Institute, and how those molecules, they are beneficial in inhibiting MIF during sepsis. The first clinical data that links MIF to sepsis was human data, which was published in 2000 by Thierry Calendra, Swiss, in Switzerland. And he showed that, like as you can see here now, this is here the level of MIF. Normally in our circulation, we have it like, this is my second talk, so you know a little bit you have to know about MIF in our circulation is around 5 nanogram per ml. And this is here where we are. And now in severe like sepsis or septic shocks, these levels could go up to from 2 folds to 50 folds. It's a diverse population. They conceive the disease indifferently, so you expect to have different level of MIF. So, but it goes up from 2 to 50 fold higher in those individuals. And this actually led the same group to ask this question. I mean, if MIF, well, it's been elevated, we know that now, whether it's been involved in the implication of or the pathogenesis of sepsis, this question can be addressed easily by neutralizing MIF in animal model of sepsis. And this what was done again. They treated animals with like, I'm not going to elaborate on the model of sepsis. It's again like everyone else here, CLP. And they give animals antibodies. You can see that those animals which received anti-MIF antibodies, like they improved survival almost up to 80% compared to the animals which received only the control antibodies, IgG, where you get only 30%. So this is the first study which links exactly MIF from in animals and human to sepsis. And what I'm going to do here, you know, I'm going to focus right now on what is this protein now. So MIF is a small protein. It's around 12.3 kilodaltons. Has been crystallized before it was even discovered it's important in sepsis. It's in 1996. 3 different studies came out showing that MIF is a homotrimer, which means like you need 3 molecules to form the crystal to form the MIF protein in the crystal structure formation. So you have a homotrimer. Each color represents subunit. Around the same time when they discovered this that this MIF has a very unique crystal structure among human proteins at that time. It was the first in its class. Now they also discovered that at the interface between those subunits here, I mean between the interface between subunit 1 and 2, there is something I'm going to call in the next couple of slides, it's called hydrophobic pockets. So it's like a bucket, like a cliff that you can fix things or you can stick things in, which, you know, this is, I'm a chemist, I'm going to try to stick things in those buckets. This is here to simplify what I'm talking about. Now, this is again, it's the MIF again, different presentations. And we only highlighted here the amino acids in yellow, which they are composing that hydrophobic buckets. And we had the hypothesis since like 1999 that if that hydrophobic bucket, if it is crucial for the MIF, what if you design a small molecule and try to stick it into that bucket and then ask the question, in the presence of MIF and the inhibitor, can you neutralize the MIF activities? And to do so, we've been involved in those chemistry. I'm going to just present them. So we have actually developed almost like 9 classes of MIF inhibitors. Each one here represents a class of at least 30 or 40 compounds. We failed in many others compounds. So we are showing you the good part of it, which is the success stories. And I'm going to focus on this molecule right now, which is the ISO1. Those molecules, which they are in yellow, those molecules, they've been tested in several animal models, in-house and in collaboration with others and independently by others. So those molecules have been tested all over, I mean, I will just say all over the world right now, I mean, especially ISO1.